students. This is chapter, 9? So the last chapter was chapter 11. This is chapter 9. So heredity is just transmitting our traits onto our next generation. So we inherit traits. Genetics itself is the study of heredity, and traits are these observable phenotypes. So once again, phenotypes, these are the expression of what our gene encodes, right? It expresses our genotype. So this should be sort of a review, but there are new concepts too. So Mendel is this monk person who ends up breeding pea plants. And he bred some purebreds, and he crossbred purebreds too. But purebreds are these pea plants that after you self-fertilize them, they'll keep on producing the same type of traits that you see, same phenotypic traits. So they're very similar in how they look with their parents and offsprings. Hybrids, on the other hand, go ahead, Diane. Yeah, no problem. Sorry, I went too fast. Thanks for raising your hand. Okay? Okay. No? You good? So purebreds, once again, if you self-fertilize these plants, they'll produce offspring that look phenotypically the same as the parents. Hybrids, on the other hand, is when you end up crossing two different types of purebreds or different types of breeds in general, and you cross-fertilize them. Definition of P generation. P stands for parental, so that's the parent generation. The F1 is the first generation of offsprings, while the F2 is the second generation of offsprings, which isn't shown on this particular slide here. So the P generation, if you self-breed them, their first generation will look the same as themselves. If you end up crossing the parental plants, as shown over here, they do cross it. Here, they're showing you that they end up using the stamens from a white plant and then fertilizing the purple plant, and then it'll end up producing offsprings that look purple, and we'll kind of go into the reason why. A monohybrid cross, this is when there's only one difference between a different organism that you're about to cross-fertilize. So if we're talking about the flower colors, this will be a monohybrid cross if the only difference is the flower colors themselves. So the P generation, if we have a purple flower cross-fertilized with a white flower, it's going to end up forming the F1 generation to be all of the purple flower itself. However, if you self-fertilize the purple in this first generation here, you'll end up having a ratio of 3 purple flowers to 1 white flower. So by looking at this, can you tell, is the purple allele dominant or recessive? Dominant, correct. Because after this first cross-breeding, we only have purple phenotype. So this is a little review. Allele, once again, is a gene that we actually have at a location of a chromosome. The dominant allele, once again, it overwrites the recessive allele phenotypically if it's like a heterozygous. A recessive allele, once again, it's not expressed phenotypically if it's heterozygous, if there's a dominant allele there. Homozygous, once again, what does it mean? It's the same type of allele that's present. While the heterozygous is different type of alleles. Yeah. Genotype is the genetic makeup. Phenotype is the Physical, yes, aspect. It's what is expressed from the genotype. So it's a physical appearance of it. And once again, what do you call this square here? Punnett square, very good. And this is the tool that we... Oh, sorry. No, it's okay. Yeah, the reason why I'm speaking, because no time. It's okay though. I don't mind slowing down. Hurry. The Punnett square is a tool that we use to help us look at the different type of possibility and frequencies of the offsprings that the parent crosses occur. And homologous chromosomes, these are chromosomes that carry the same types of alleles. So they don't carry the exact same alleles, but they do carry the same types of alleles. No. The name is the same. Homozygous, when we talk about it, the homo is the same. So it's the same allele. This is homologous chromosomes is the same type of chromosome. Okay, so remember the sex chromosome for females is XX, right? And those are homologous chromosomes. That X is homologous with that other X. Even if it's a male, right? XY, the X is a homologous chromosome to the Y, although they're not the same shape or... So, technically speaking, I guess they're not really homologous because they don't share the same genes. But all the other 22 chromosomes beyond that, the 22 pairs, when they pair up, those are homologous chromosomes. A gene locus is one location of a chromosome. So just this location or this location or this location. And these are where the alleles are located at. So we talked about monohybrid cross, right? So that's a difference in only one aspect, right? If we're talking about dihybrid cross, now how many aspects are there? Two different aspects. So, for example, we have this Punnett square where it shows different types of aspects. One is color of the seeds themselves, and the other is textures of the seeds. So, color-wise, it could be yellow or green. Texture, it could either be wrinkled or round. This is the round ones. These are the wrinkled ones. So in this here, we can tell that the yellow and round peas are the ones that are dominant alleles, while the wrinkled and green ones are the recessive alleles. There are these two Mendel's laws. One is law of segregation. Law of segregation is that the gamete will only receive one allele or the other, meaning that the gamete will either receive an allele from the father's side or the allele from the mother's side. Law of independent assortment, on the other hand, is very similar to what we were talking about during meiosis 1, right? Where it's the random assortment portion of it. So each pair of alleles, they'll segregate independently from each other when they end up forming their gametes. Do you guys remember which stage of meiosis 1 this occurs? Yeah, anaphase 1 or metaphase 1, depending on how you describe it. Jessica, you had a question, or you're just raising your hand?
Oh, no.
Oh. Okay. Oh, yeah. Okay. Yeah. This is where it occurs. Metaphase 1, anaphase 1 for this independent assortment. A test cross will help you determine whether or not dominant phenotype is actually a homozygous dominant genotype or a heterozygous genotype. And you do this by crossing that dominant phenotype with a recessive phenotype. So here we have a Labrador here that's black, but we don't know whether or not this black Labrador is homozygous dominant or heterozygous. So what we do is we cross it with a recessive individual. This recessive tends to be more chocolate type of color or brown color. And so when we cross it, then the outcomes will be, if we end up crossing these two, if it's heterozygous, we'll end up having some offspring that will end up becoming brown, while others are black. However, if it was homozygous dominant, what kind of offsprings will we have? Just black. So here's another example. If a parent flower is purple flower pea plant, what is the parent genotype if we end up crossing it with a test cross and their offspring becomes all purple flowers? What is the parent genotype? If we're just saying, okay, let's say big P is purple allele, for purple flower allele. So what is the parent genotype then if the offspring of the test cross produced all purple flowers? Homozygous dominant, very good. So it would be big P, big P.
Even if they were both heterozygous, though, wouldn't they be able to produce a purple flower anyway?
Yeah, it will still produce purple flowers, but if you have multiple offspring, you'll have a 3-to-1 ratio of having, or not 3-to-1, it's actually half. So if it was heterozygous, just say, we cross it with homozygous recessive, and you do that Punnett square, right? So this is going to be a big P, this is small p, this is the recessive one, little p, little p. So this one will be big P, little p, big P, little p, little p, little p, little p, little p. And so you have half of them becoming purple flowers, the other half will become white flowers. Okay, we just answered this question, right? If the offspring had half of them being purple, half of them being white, then the parent genotype would be heterozygous. Very good. The rule of multiplication, this is having the probability of multiple events occurring. You end up doing this by multiplying each independent event. So when they're talking about this, let's just say, what is the probability of having two flip coins having both of them being heads? Okay, what's the probability of getting heads or tails? 50%, right? So half, right? If you want to get two in a row, heads up, and two in a row, then you're going to end up multiplying it again. So it's half probability of getting heads up the first time, half again to get a heads the second time, so you end up getting 50%. 25%, 1/4, right? Which equals 25% probability of getting two heads in a row. What's the probability of getting heads and a tail in a row? So how would you calculate it out? How many percentage to get a head the first time? 50%. How about just the tails the second time? It's also half, right? The tails the second time. So you end up getting 1/4%. Does that make sense? How about getting tails the first time and then heads the second time? Same thing. Okay. Yeah, you guys get the pattern, right? Yeah. That's why it's a little tricky. I mean, if you go to Vegas and stuff and you're going to roulette, I heard that there's a sign that shows you how many times it's been black, how many times it's been red. And it's been a long time since I've been to Vegas. but but I heard that there's a sign, and so basically they're tempting you. If you see like 10 times that it's been black, 10 times, where are you going to place your bet on? Possibly black, possibly red. But what is the probability of the next one being red or black? It's still 50%. Actually, it's less than 50 because it has that 0 and double 0 or something like that. So it's less than 50%. Agreeing. Thank you. I'm not a gambling person. So, let's see. We'll look at that later. Okay. But family pedigrees, this is where genetic information that we know about a family and their family tree. And we form a family tree using these square symbols here for males, while circle are for females. Whenever it's shaded like this, it's actually the particular trait we're actually looking at. So, for example, first of all, the ones that are usually nearer to the top are the first generations or the oldest generations, while the ones nearer to the bottom is the younger generations. Looking at this particular one, this is just looking at the different types of earlobes, whether it's attached or detached. If it's attached, then it's the one that are the homozygous recessive. So these are the recessive phenotypical traits, while the ones that are detached are the more dominant phenotype. So here you see that this recessive genotype will end up causing their daughter, in this case, to have that particular trait. While this one is a carrier, right? It has that particular allele, but it's masked by it being heterozygous. When you make a heterozygous with a heterozygous, then there's a possibility of having children that has the recessive trait. And do you guys remember that probability if it's a heterozygous versus heterozygous? Oops, sorry. If it's a heterozygous mated with another heterozygous, what's the probability of having a homozygous recessive child? 1 out of 4. Yeah, this one here is homozygous dominant. This is heterozygous. This here is also heterozygous. This is the homozygous recessive. So it's 25%. Okay, so what is the probability of getting a male, Kevin here, a male and also having a homozygous recessive trait? So we already calculated this, right? This was how much? What's 25%, right? 1/4, right? What's the percentage of getting a male? One half, right? You're either going to be a male or a female. So 1/2. So the probability of getting a male offspring that has this attached earlobe is going to be 1/8, which is, no, it's not 20%. It's less than that. 1.6 something, or 125.1. 12.5, thanks. Okay, so now we're going to get a little bit more complex. So it's not just straight dominant and recessive alleles. We've got this thing called incomplete dominance. And incomplete dominance is when you have two different purebred parents. If you end up crossing them to produce an offspring, the phenotype of it is actually a combination between the two parents. We talked about earlier on with the pea plants where it's purple and white, and they produce offsprings that is purple. In this particular case, when we're talking about the snapdragons, this red snapdragon, when mated with this homozygous recessive white snapdragon, will end up producing offsprings that are pink. So the heterozygous for these plants are a combination of the two different colors. So when you make this, if you self-fertilize this pink Snapdragon, what would you end up having then? Yeah, if you self-fertilize the pink Snapdragon, so this is the heterozygous, right? What is it going to mate with? Yeah, so same thing, R, big R, small r. So you're going to end up having homozygous dominant. What color is that? Red. Very good. So 1/4 would be red. What's this one down here? Pink, yes. It'll be heterozygous. Pink as well. This and a white one, yes. This is similar to this thing called hypercholesterolemia. This is when you have this extremely amount of high cholesterol within your blood. It is a recessive allele trait, and so the thing about this is it has an increased amount of chances of blocking arteries to cause heart attacks and stuff at an earlier age. People with the dominant alleles are normal. Those with heterozygous, as shown over here, will lack these receptors. These receptors take up the cholesterol and allowing it to be digested by the liver later on. However, those with lesser amounts of these receptors will not take up the cholesterol, so it stays in our blood for a lot longer. So the heterozygous tends to have 2 times the amount of cholesterol levels within the blood. And then those with recessive, these will lack a lot less, or lack these receptors to bind cholesterol, and then so they tend to have even more higher concentration of cholesterol in their blood. So these also potentially could have heart attacks at the age of 2. Yes, because they lack of these alleles to absorb. Eventually, the cholesterol gets absorbed or gets broken down by the body. Without those receptors, then it stays in the blood, blocking the blood vessels. So that was codominance, not codominance, incomplete dominance. Codominance is a little different from incomplete dominance. This is when both alleles are actually expressed, even in the heterozygous individual. So our blood type is an example of this. We have multiple alleles, and then these alleles, there are more than just 2 alleles. We have the A, B, AB, and O blood types. And remember how the blood types help determine the antigens, the glycoproteins that are on the surface of our erythrocytes. And that's what determines our blood type. And these are the alleles. We have these types of alleles. We have the immunoglobulin A, immunoglobulin B, and then just the lowercase immunoglobulin. And so for those that are blood type A, how would you write them? If it's homozygous dominant, it would be IA, right? If it's heterozygous for this type blood A, how would you write it? IA and lowercase I. Okay, so this is both type A. Okay, for type B then, tell me. IB, 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 lowercase I. This is for type B. How about type AB? IA, IB. Any other ones? No, it's just AB. And then, no, there's only 2 alleles. So for type O is going to be 2 lowercase I. Yes, I can. Yeah. Okay, so we just named the different type of genotypes and phenotypes. So this is just to show you the different type of genotypes. We got that correct. All these we got correct, so very good. And if it has that type of genotype, remember, it gets transcribed and then translated so that we produce these antigens on the surface of the erythrocytes. And this is just showing you the different types of antigens that we have. And AB has a combination of both A and B antigens, and that's why you have these triangles and circles. The circles are for the antigens for B, the triangles for the antigens for A. And O, type O, doesn't have any of these antigens. Yeah, it has that genotype, but I'm saying there's no, it's different, the phenotype, yeah. And this is just showing the reaction when you have different blood types mixed with other blood types. And this is when the antibodies end up binding or clustering around the red blood cells that have that attaches to that particular antibody. And that's why you can't give blood type B to a blood type A person, because it's going to cause this process called agglutination. And if you do, agglutination will possibly block some blood vessels. It's not really a clot, but it just blocks the blood vessel, kind of similar to a clot, though. Yeah. Okay. Okay, so one of the assignments is for you guys to actually figure out your blood type, if you can. If you know your parents' blood type, so you gotta ask your parent what blood type they are. And then if you know some of your siblings' blood type, then it will help narrow you down as to what blood type that you have. If your parents do not know their blood type, ask your siblings if they know. If they know, then you could potentially project what your parents' blood types are. If you don't know any of that information, just make up your parents' blood types and then figure out what potential blood type you will be if your parents are like that. Okay? Pleiotropy. This is when you have one gene that ends up coding and causing several different aspects because of that one gene. For example, sickle cell disease. Sickle cell disease is actually a codominant allele, so it will end up causing several different complications. This one allele causes, well, first of all, the erythrocyte from forming normally. So it causes the sickle shape to form, and that will cause, due to a lack of oxygen, it will form this sickle shape, and then it might end up causing the erythrocytes to be destroyed themselves, and it could potentially clog capillaries as well because of its abnormal shape. And due to that fact, when you have not enough erythrocytes, it causes anemia, not enough oxygen within your blood. It also could cause pain, fevers, and other types of damage as well. So this is just showing you that this one allele with sickle cell disease can cause several different types of outcomes due to that one allele. So this is pleiotropy, an example of it. Just all different. If you end up causing not enough blood to, just say, your arm, you gotta possibly amputate it, worst case scenario type of thing. That kind of damage. Wherever it doesn't have enough perfusion of blood or enough oxygen in this case to your heart, it will cause a heart attack. To your brain, cause a stroke, etc. Okay, polygenic inheritance. This is the exact opposite of pleiotropy. This is when you have multiple genes that will all cause only one single phenotype, while the other one was one particular gene that causes multiple different types of outcomes. So here, this is typically thought of as being only one particular gene. So this is not a polygenetic inheritance. This is only one particular trait or gene that codes for freckles or codes for widow peaks. Widow peaks is this portion of it where you kind of look like Mickey Mouse kind of thing. And then we already talked about the free earlobe or detached earlobe or attached earlobe as well. So these are all single trait or single allele, or what they think that's a single allele that will determine these traits. Polygenic inheritance, on the other hand, height is an example of a polygenic inheritance, meaning that there's several different types of alleles or genes that will end up making up our height. So this is just showing you, in general, we have this bell-shaped curve, so very few of us are going to be very small, and very few of us that will be very tall, height-wise. I think it depends on nationally as well, like because of height is also controlled by what we eat, diet-wise, and exercise. So our culture actually plays a role in it, as well as genes. Yeah, I mean, if you're malnutrition, you're not going to grow as tall. Yeah, I think. Yeah. Yeah. So environmental factor, like I said, is a key factor of phenotypes as well. So like this is genetic twins, where they do look kind of different though, due to different type of how they were raised and whatnot. So these environmental factors are actually not passed down to the next generation. And we already said that height is not only affected by genotype, but also affected by this environmental factor. And this is the whole debate of nature versus nurture. Nature meaning our genes that will cause our development. Nurture meaning that our environment factors, whether our diet, our exercise, different types of environmental factors, the amount of they're different physically. I mean, yeah, the twins here. Yeah, I mean, if you were to weigh them, even they might be different weight as well. But I mean, like even by appearances, they might end up looking a little different as well. Like this one has like a mole here or something. I don't know if the other one has one. It doesn't seem like it. Maybe it's the angle of it, but or the makeup or whatnot. But what I'm trying to say is like even their height might differ as well, depending on like what they've eaten, how much exercise they've gotten, and stuff like that. There are different types of human disorders. One is autosomal recessive disorders. And these autosomal recessive disorders, this is, like the name implies, it's a recessive disorder. And so those that are heterozygous parents are carriers. They carry this recessive disorder. So most of the genetic disorders are these autosomal recessive disorders. There's these different types. Or I guess the only single type that I have great, cystic fibrosis. We'll talk about that. There are other autosomal dominant disorders, and this is different types that we'll talk about: polydactyly, achondroplasia, and Huntington's disease. And we'll go into each. This is achondroplasia. Achondroplasia. Yeah. Well, one type. Yeah, I'll explain them. So the autosomic recessive disorder that we'll talk about is cystic fibrosis. It's actually very common in the U.S. actually. This is basically a genetic disorder that causes a defect on the chloride transporters. And if you guys remember, like chloride, these chloride transporters will help pump out chloride into the outside of the cell. That's where most of the chloride is supposed to be located at. However, these transporters are not functioning within people who have cystic fibrosis. And so what happens is that there will be a buildup of mucus wherever it might be. If it's in the lung, there will be a buildup of mucus within the lungs. And unfortunately, that will hinder them from transporting oxygen and transporting carbon dioxide out, gas exchange. So these tend to have different type of treatments. One is by pounding their chest. If they pound their chest, they're going to loosen that mucus to try to cough it up. Clearing their lungs, they have devices that actually try to suction the mucus. So it's very painful. They also have to change their diet and also different types of antibiotics to prevent any kind of infections that occurs. However, they tend to still have a shortened lifespan. Inbreeding is mating between people who are closely related. The reason why I bring this up is if someone has a recessive genetic disorder, they have more chances of producing offspring that will have that genetic disorder. So if you have two carriers, two heterozygous for that genetic disorder, their offspring, the potential will have an offspring that is homozygous recessive, to have that particular.
There's other risk factors with inbreeding other than
Genetic
disease, right?
I think that's a major thing. I don't know anything else because usually it's the genetics that I mean, culturally it's kind of not looked upon well, right? But it's, I mean, if we're talking biologically speaking, there's these chances of these recessive disorders to occur. Say again? Cloning humans, it's different because when you're cloning humans, you're using the exact same genes that you currently have. So if you currently have these recessive disorder and you clone yourself, that clone will also have this recessive disorder. But if you don't have it and you're just a carrier, your clone will just be a carrier as well.
So inbreeding is always wrong as humans, but dogs or like animals.
Same thing. So it's the same thing. That's why.
Are purebreds better than?
Not necessarily. That's why these purebreds, that Labrador I showed you earlier on, the Labradors, they have a recessive allele where they cause them to be blind. And so there's a percentage of Labradors that will be blind because of all that inbreeding occurring. As for autosomic dominant disorders, polydactyly, this is when individuals will have extra digits, whether it's on their hands or on their toes. The achondroplasia, this is dwarfism, one type of dwarfism, where the head and torso still has normal development, but their appendages, arms and legs, are shorter. Once again, those that are homozygous dominant will die off, so those that are heterozygous are the ones that end up, it should be not the bottom. Those that are heterozygous will be the ones that has this phenotype. However, those that are homozygous recessive, like this individual and this individual here, will still have a normal height. Huntington's disease is also another dominant disorder. This is a degenerative disease of the nervous system. However, since it begins in middle age, some people don't realize this, and so they end up passing on their genes to their offspring before realizing that they have this particular disease. And it is fatal to eventually, so these people don't end up living that long.
So you know how people are pregnant and they take the fluid from the embryo? Like, do they test for these things or no?
They can potentially test for these things, yes. Which we'll actually go into really soon. Genetic testing. So genetic testing, they help detect these diseases caused by these alleles during pregnancy. The thing about this is that once again, it's an ethical kind of issue because once you do detect that there is these disorders within your offspring, usually they would want you guys to have counseling to kind of understand the disorder or just understand even this process of genetic testing to figure out what types of ways can we help this child who end up having this, getting born with this disorder and stuff like that. So counseling is involved as to help the parents cope with and see how they can be able to live with the child. The ethics that are included in here is like, if you do know it at a point in time, then whether or not to go on with the pregnancy or not type of thing. That's where the ethics comes in. I don't think so. I think that's why it's the controversial thing.
So you can know whether or not your child's going to have
A particular disorder. Yeah, I believe so. Yes. Yeah. Usually, I would say very high, close to 100, basically, unless they get like cells that happens to not be the embryonic cells, because those embryonic cells are the ones that will have the complete DNA already, and there are certain, unless it's something that we haven't discovered the coding to, kind of thing. Like if it's a particular disorder that we don't have a sequence of DNA yet, then we don't know if that disorder is in the child or not, because we don't know that sequence. But if we do know that sequence, we could look at the DNA sequence and just pinpoint where if they have it or not. Yeah, if they do have it, then they have it. Okay. If they don't, they don't. Type of thing. Okay, linked genes and crossing over. So linked genes themselves are genes that are very close on the same chromosome, that are very closely located near each other. If they are located close to, okay, so my example is a dihybrid cross, right? So we have, or basically kind of like a test cross, right? Where we have a heterozygous of this dihybrid cross mated with a homozygous recessive. Okay, so what kind of offspring will you think this person or these flies will have? What kind of genotypes? So so we have, you have this genotype here, you have this genotype, you have this genotype, and this genotype for this parent here. For this guy here, it's just going to be this genotype, right? All the combination will just be lowercase g, lowercase l. So this individual here will be heterozygous for both, right? This individual, heterozygous for only the body, gray body, but recessive for the wings. And over here, we're going to say 2 lowercase, right? And then 1 large. Yeah. So this one here will be very similar, short wings and black body here, right? So that's what our predicted one. However, if they are linked together, you'll notice that you'll have some, or majority of them, to be these two here, which we did predict as some of the outcomes. But you'll notice that also that very few of them will be these ones here. So originally, what was the percentage here? It would be 25% of all of them, right? We should see equal amounts on each of these genotypes if they produced it. But you notice how over here we have abnormally amounts of this one and this one here, but very few of these one. And this is due to because they're linked together. So they don't undergo independent assortment like we normally think of it. And this is due partially because of crossing over. If they're very close together, they're not going to cross over as frequently. So notice how over here the crossover does occur within these gametes here. So if we're talking about these two alleles here, if we say that, let me redraw it. if the alleles actually were at another location, and I said that one of them is over here and the other one's over here right next to it, then crossing over with another individual or another homologous chromosome, it's not going to happen as readily, right? If they were further apart, if instead of them being this close, if they were like over here and over here, then crossing over will have more chances of occurring. However, if you're just talking about just this small location that, oh man, this one small little location here between these two areas, then crossing over, there's less chances for them to cross over, and that's what makes them kind of linked together. Does that make sense? No? Yes? So over here, they're far enough apart that crossing over can occur. If they were closer together, if this B allele were over here next to it, the crossing over occurred at this location, right? But if they were right next to each other, even though there's crossing over occurring, will these two alleles be on different chromosomes? No, because it will cross over at this location here, away from it, so it'll still be linked together on the same chromosome. Okay, and that's what's causing this to occur. This is linked together with the G is linked together, really closely related with the L's, basically. So if you instead of A and B, you're thinking of that G and L closely related together. And this is just showing it to you. So since they're close enough, most of them will end up becoming like these ones here. But sorry, these two here. But these two here. But you notice how the amount of crossing over that occurs is not that much. And that's why we have fewer amounts of these. Because they're so closely together in location. So this linkage map is kind of related to that. This is when we end up mapping genes just due to the amount of probability of crossing over that occurs. So if there is more crossing over, we know that these genes are further apart from each other. However, if these genes, if there's less crossing over occurring, then we know that the genes are closer in proximity to each other because crossing over won't occur as frequent. So let me just go to that example again that I've been telling you. So this GCL, okay, if we say that G is over here, and let's say C is over here and L is over here, okay, the chances of crossing over for G compared to C and L is more, right? Because it's further away from these two genes over here. So if I had another homologous chromosome over here, then this G over here will most likely cross over, will have more chances to cross over in comparison between the C and L. So what I'm trying to get at is that if crossing over, let's say crossing over occurs at this location. If it does occur at this location, then this red G over here will be over here, and the blue one will be on the other side, correct? Okay. But notice how this crossing over at this location doesn't affect the crossing. Doesn't affect the. There is no crossing over on the CL location because it occurred at this location. If we say that crossing over can occur at any place on this particular chromosome, then there's a lot more chances of crossing over G compared to C and L because there's all these locations where crossing over of G occurs, right? However, the amount of locations of C and L crossing over will be less likely to occur because there's only these locations here that L can cross over, but C doesn't. Okay, because they're so closer together that the chances of crossing over doesn't occur as much. Yeah. Okay, the closer that the genes are located at, the less likely they are to cross over. Okay, this is a review. Autosomes, these are non-sex chromosomes. And sex chromosomes is the one that determines our sex. So it's our 23rd pair of chromosomes that determines our sex. Males, once again, are what? XY. Females are XX. XY, the Y itself determines the male sex. It's the males that determine whether or not it's going to have a male or female offspring. Since males only have the Y chromosome. Sex-linked genes, these are genes when we're talking about that are only on the sex chromosomes themselves. So most of them are going to be on the X chromosome because they're so much larger in compared to the Y chromosome. However, there are some genes on the Y chromosome, not many, about 12. One of the sex-linked disorders is red-green color blindness. This is causing a mutation on the cones. Remember the cones? What are the cones for? It's within our eyes that detect color. Very good. If there is a mutation on them, then you're going to prevent them from receiving normal color perception. And that's why it mostly affects males compared to females, because it's on the X chromosomes themselves. If it's on the X chromosome, you can see over here where we have a normal female with crossed with red-green color blindness male. We end up having carriers for females, but normal males. When the carriers mate with a male, normal male, then we potentially will have someone who actually expresses color blindness male. However, the female, notice how the female is heterozygous. If it's heterozygous, they will have normal vision for the most part. And so they are mostly carriers, so it occurs more frequently in males than females. And so this is just showing the red-green color blindness. For those of you who cannot see it, that means you do have this red-green color blindness. For those of you who can see it, it means that you're normal vision. 7. Are you being serious, or are you just messing? Hemophilia. Hemophilia is also a sex-linked recessive trait. It does cause a defect in the protein factor that causes blood clots. And we already talked about this before. So if you have a disorder in this, you potentially could bleed to death. So Queen Victoria was a carrier of the hemophilia, and she introduced this to the different royal families, and this one is from Russia. And so we were talking about inbreeding earlier on and stuff with the royal family. If you end up only reproducing with other royal families of other nations and stuff like that, and just keep it amongst yourself in that sense, then you have a smaller sample size that will cause this allele or gene to occur more frequently than in the normal population where it doesn't happen as frequently. Acknowledgements, pictures, we're done.